Dr. Andreas Wagner from Klostenburg, Austria.
Thank you. Thanks to the organizers for inviting me and giving me the opportunity of presenting some of our efforts, our capabilities. Yeah, Polymun Scientific is a fully private company located in Klosterneuburg, nearby Vienna. And what we are doing, we are CMO on the one side for biopharmaceuticals, here mainly mammalian cell culture, and the other part, which I represent here, is the liposomal formulation part. Here are some selected reference projects in this field. As you can see, we are on also for drug delivery as well as vaccine formulations. For drug delivery, we are mainly working in the field of oligonucleotides, but we also work with small molecular weight drugs like curcumin. And one highlight here, where we have heard an excellent talk yesterday, is the process that we do with Pronide Therapeutics, where we have heard yesterday the excellent progress in their clinical trials with a DNA eye drug encapsulated in liposomes in our lab. When we speak about liposomes from a CMC point of view, it's not that just take out lipids out of the freezer, mix it, and then inject it into animals or later on into patients. We have to take care about raw materials, about qualities. I come to this later on, the formulation procedure, and the refining process. All this has to be embedded in a good analytical QC background, which by itself has to be embedded with all the other points in a well-established and quality systems where all the people in the company can live with, also in and external the company. So when we speak about lipid quality, in the meantime, the lipid supplier managed to really produce excellent lipid quality, and they also support us in some cases with analytics. And this is a very important point right from the beginning when you start with lipids, liposomes, to determine the raw materials that you use, that you can also determine byproducts or quality of this material right from the beginning. For the chemical and physiological properties, I don't have to tell you too much. You can choose lipids which have a pH sensitivity, fusogenic activity, and so on. The same is with API. Take care about API quality, purity, microbial load, and endotoxin content is very important, because when you have endotoxin in your product and you formulate this in liposomes, you will never get rid of this endotoxin. So they will entrap into the membrane and therefore work with clean and good material right from the beginning. For the API also, you have to take care about the stability during processes and also during storage. API solubility is a very important point, because this is important at which stage API comes into the process to get encapsulated in liposomes. When you speak about preparation or production techniques, this is a list of methods which you find in the literature. Some of them are mild methods, some of them are more invasive methods, and these invasive methods lead to lipid degradation or API degradation throughout processing. Therefore, you can either choose these methods and move on with helper substances, or with with lipids which you choose, that this oxidation or hydrolyzation can be minimized. Or what you can also do, you can work with rather mild procedures. One of them is the film method or the ethanol injection. We, by ourselves, found that the ethanol injection is the most usable, especially when we speak about scale-up. This is how we realized this scale-up. We replaced steering of the aqueous phase by pumping, and by this we pump the aqueous phase from one to a second vessel, and during this we mix the lipid ethanol with this aqueous phase, and here liposomes are formed. We can control the liposome size and homogeneity quite well with these parameters that you see here. All these parameters well defined by this mixing step in this process, and thereby in most, I would say 95% of our processes, there is no downsizing step needed. So there's just, this is a one-step procedure for the liposome formation, followed by a filtration process to remove the ethanol and unentrapped API. What are the advantages of our systems that we have developed? It's fully scalable. We can produce under aseptic process condition. What you see here, it's a closed containment which can be sterilized, sanitized, and all raw material can be transferred into the system via 0.2 micron filtration barriers. This is a picture of small-scale experiments in the lab. I like this yellow liposome suspension, which is related to curcumin and trapped within the membranes. But once we have determined these parameters at a very small scale, 50 to 100 milliliter minimum, we can easily transfer this into the large scale. This is a picture from our GMP unit where we work with movable tanks. Currently, we can produce up to 250 liter liposomes within one process per day. This brings me to the next point that I mentioned before. This is a list of equipment which shows you that we have a mixture of single-use as well as fixed installed equipment. By this, we can handle these volumes quite well. But nevertheless, we are currently planning, due to the progress of at least one of our customers' projects, to build a new facility where we work on liposome productions in cubic meter scale. This is a picture from a process in one of our labs that you can see how liposomes can be produced in the industrial scale and not just in the lab. These are pumps and vessels that we use. What means GMP production of liposomes? Sometimes people come to us and ask, can you make me just 500 milliliter of liposomes, that it's not too expensive? We just need it for first human trials. But it's not the volume which counts, it's all the QA, QC work in the background which counts and which makes GMP production more expensive than early research work. You have to set up all these cleaning programs, the hygiene program, handling of materials for steam sterilization, and so on. All this has to be defined, and all these QA defined systems have to be reproducibly done every day to keep the high quality of the system. This is just an overview of what you have to think of when you go for GMP production. For this technology that we have established, we can encapsulate all different kinds of APIs by pure passive encapsulation, for instance. We have also established a one-step active loading. Substances like Doxil can be encapsulated like this by a pH gradient. What we also do is an advanced loading of oligonucleotide drugs, and we can also encapsulate membrane-associated substances. Here are some examples. Here is, for instance, for DAFRA, we have established OLPC-containing liposomes. This is a non-phospholipid vesicle where the API is encapsulated within the membranes. This process was transferred from the Max Planck Institute into our labs, and we have set up the GMP production for this. Signpath Pharma, what I mentioned before, is curcumin within liposomes, and this was a vaccine project which we finished this year on a European grant basis. Finally, it's a very homogeneous method where we get very good batch-to-batch consistency. Our products that we produce are stable over years, even when we store it in suspension, not lyophilized. And it's a very clean process where the raw materials can be sterilized at the end of the day by 0.2 micron filtration. So finally, what I wanted to show you is that you can successfully produce liposomes on a large scale for all different kinds of APIs. It's feasible to do it in a GMP-compliant manner and you can scale it up. As I mentioned before, now we plan for a cubic meter scale. Thank you very much, and these are some reference projects.
Thank you very much for the very comprehensive presentation. And now the floor belongs to the next, also a company representative.